All right, so we are starting chapter 7 today. We're on chapter 7, section 1. And all of chapter 7 is all about enzyme mechanisms, so how enzymes work. And you got to know that enzymes are used constantly by our bodies. I'll give you one example talking about cholesterol, right? We know that we need cholesterol inside of our cell membranes, but if we eat foods that are really, really rich in cholesterol, your book shows egg yolks, red meat, that kind of stuff, you're going to end up with really high serum cholesterol levels. So serum meaning in our blood, right? And we know that if we have high serum cholesterol levels, that correlates with arterial blockages, with cardiovascular disease, all of that kind of stuff. So we give people medicine, and those medicines are called statins. You've probably heard that before. But what do statins do? Well, they inhibit an enzyme. Imagine that. They inhibit an enzyme called 3-hydroxy-3-methyl-glutarate coenzyme A, HMG-CoA, reductase. So it's an enzyme, it ends in ase, it's a reductase. And so this is one of the enzymes that catalyzes cholesterol synthesis kind of in the very beginning of the process. And so if we inhibit this one enzyme, we can decrease synthesis of cholesterol in our bodies. And that's really important because then the body's going to say, well, I can't make it, so I'm going to uptake it from my serum. And so the cholesterol leaves the serum and gets used instead of being circulating and therefore being bad. So we can use our knowledge about enzymes to do really, really pretty cool things. So we're going to do a quick overview of enzymes today, and then we'll talk about structure, function, how they work in reaction mechanisms. We'll talk about kinetics, and then how we regulate enzyme activity. So biological reactions are super slow. And enzymes are what allow these reactions to happen faster under our normal physiological conditions. Remember, we can't increase our temperature to speed up a reaction or we'll die. So it's important that we have these enzymes to speed things up, but it's also really important that we regulate these enzymes very tightly. Because you know that enzymes are not consumed in a reaction, right? They don't change what they are, so we have to regulate them. And another important reminder is that they do not change the equilibrium concentration ratios of reactants and products, right? So that's just your quick enzyme review, which you should have gotten in Chem 10 something. All right, so remember, enzymes can really be proteins or ribosomes, but we're only going to talk about them as proteins in this chapter. There's a whole chapter that's devoted to RNA, enzymes, ribosomes in chapter 21. Okay, so it all started in about 1926, and James Sumner was the one who first figured out that enzymes are actually proteins, right? People had been studying enzymes for a long time, but they hadn't made that jump that enzymes are actually proteins. And so he first purified and crystallized urease. That was our first enzyme. And then pretty soon after that, other people got on the train, and pepsin, the stomach enzyme, was discovered next. And so those two guys shared the Nobel Prize in, I think it was 1946, for protein purification and characterization. So what we really know and understand about enzymes really relies on the 3D protein shape or structure. So this is super important. So when we first started looking at enzymes, we kind of thought, oh, it's a lock and key, right? Your key and your lock never change shape. They fit inside, the key fits inside the lock, but the lock can kind of tumble, but the lock is always one particular structure and the key is always one particular structure. So this is kind of an old idea that assumes that we have a rigid physical, chemical complementarity between our enzyme and our substrate. So really pretty fixed. So that substrate binds to our enzyme just perfectly. And this is really old and not not what we understand now. So the induced fit, they started with this in, I think it's almost 1960, where we started to understand that enzymes are flexible and they accommodate this substrate by changing shape, right? So the substrate doesn't really fit until it enters the enzyme, and then the enzyme changes shape. So this allows for a whole host of weaker interactions that are going to facilitate that substrate enzyme interaction. So it's kind of analogous to an enzyme, instead of being a lock and key, right? Maybe it's a hand and glove. So the enzyme is our really flexible glove, and it fits on an equally flexible hand. So both the enzyme and the substrate are flexible. So that's kind of an important thing to kind of take home. So there are some critical aspects of enzyme structure and function that we have to understand. And the first one is that enzymes usually bind to their substrates with very high affinity and very high specificity. So this example here, this enzyme is phosphoglycerate kinase, and it transfers the phosphate group. That's its job, right? And this green area here, this is called the binding pocket or the active site. So either way you want to say it, it's the same thing. And this is where the substrate's going to bind, and then you're going to promote catalytic reaction. So how do you promote a catalytic reaction? How does that happen? So the active site is this protective environment for the substrate, right? So it can get away from the solvent. So that substrate can bind to our enzyme, fits into the pocket or into the active site, and that enzyme's functional groups within that active site can then interact with the substrate. So here it's showing you the arginine residues, 1, 2, 3, are able to interact with the substrate. And it's not the only ones that are interacting here, but it's a good illustration of that. All right. So the other critical aspect of enzyme function is that our substrate binding to the active site induces changes. So this was that really big jump, right? So because of this, we have all these weak interactions that are going to be able to happen, like hydrogen bonding, ionic interactions, van der Waals, right? All of those kinds of interactions are going to facilitate the structural changes that are needed to form the products. So we're going to have conformational changes. This is a really nice example of the conformational changes. This is hexokinase. It's a metabolic enzyme. And so glucose is what fits inside the active site. And so this is a huge conformational change going from free hexokinase to hexokinase that has our glucose bound in that center pocket. And so you have this insane kind of folding in of the enzyme. So usually changes are more subtle than this, but this is a really nice illustration of how those conformational changes can actually block the solvent. They can block water. They can keep the solvent out to facilitate the reaction, like phosphorylation in this case. So I guess I showed it twice. Okay, so this is the illustration of our induced fit model, right? And that's exactly what we said, that the glucose molecules here, when it fits in, you have a massive conformational change. I guess I kind of put that twice. So the other thing is, not only does glucose fit in, but here's the unbound hexokinase, right? And water can very easily go into the active site in hexokinase. So this is exactly the same molecule. In here we were showing space filling. In here we're showing ribbon diagram, but it's the same molecule. And so what we see is that water can very easily get into this pocket. But once glucose is bound inside of the substrate's active site, now you basically had the molecule fold in and protect that glucose molecule from being exposed to the solvent to allow it to actually be phosphorylated and catalyze the enzymatic reaction. All right, the third one is our enzyme activity is very highly regulated in cells, right? We need to maximize that energy balance between catabolic and anabolic pathways. That is highly, highly regulated. And so there are multiple modes of enzyme regulation. So there's bioavailability. This is the first one. And this is where you control the amount of the enzyme present. And there are two ways that you can do this. You can do this through gene expression, right? You can transcribe it more to make more. You can transcribe it less to have less. Or this can be done through protein turnover. So how quickly you degrade the protein. Either one results in a change in bioavailability. The next one is catalytic efficiency. So this is how we control enzyme activity. So you're not changing how much the enzyme is there, but you're changing how active the enzyme is. So this can be controlled by binding of regulatory molecules. And it's very similar to, like when we were talking about hemoglobin and how the 2,3-BPG binds to it, induces conformational changes, and now it can't bind O2 as well, right? Similar to that. So this is binding of regulatory molecules. That's one. The other is by covalent modifications. So when we talk about covalent modifications, do you remember when we were talking about amino acids? And the amino acids, serine, threonine, and tyrosine. These were a special kind of grouping because these three can be phosphorylated. So phosphorylation of serine, threonine, and tyrosine can all result in regulation of our enzyme of interest. And then also non-covalent, binding of AMP. Binding, right? So either one of those two are going to change usually the conformation of the molecule and allow the enzyme to work. So it can bind, either one of these can be close to the active site of the protein and very easily change the conformation, or it can actually be on a distant part of the molecule and you can have a propagated conformational change. So you can have a distant effect, which is kind of cool. That it can be far away and still have an effect on the enzyme activity. All right, so how do enzymes work? They are chemical catalysts, right? And we know that chemical catalysts can alter the rate of a reaction, but they do not change the ratio of substrates and products at equilibrium. So if we have kind of our equilibrium expression, A is going to B, but it's reversible, right? And so we can decrease the activation energy of our reaction in order to speed it up. So we're reducing the amount of time it takes to reach equilibrium. Our catalyst is going to not be specific. It can increase the rate of a reaction in both directions by the same amount. So it can increase A going to B, or B going to A, either way, right? So while our rates are affected, the final equilibrium and the overall delta G are unchanged, because you have to remember what delta G is, right? So remember, delta G is our change from the energy in our ground state of our reactant, or whatever we really were, in order to get to the ground state of the product. So if you have a catalyst, those two things are not going to change, right? But what you have to remember is that when you're going from reactants to products, that substrate is converted into a high energy transition molecule. And so that's indicated by this right here. And this, they call this the transition state. So anytime we talk about things in the transition state, we use this symbol, and this symbol is referred to as a double dagger. It sounds very ominous. But so this transition molecule can either become a product, or it can stay a substrate. It's super unstable. It's very high energy, but very, very, very unstable. So you only really have a few molecules at a time that are in this transition state. And so what happens is we can look at a comparison of an uncatalyzed reaction to a catalyzed. So the uncatalyzed is our red and the green is the catalyzed. So if you look at the change in energy to get to that transition state for our red reaction, I guess I should have done that in red. For our red reaction, what do we see? This is pretty big. So this is EA, or activation energy, EA, of our uncatalyzed reaction. And so that's pretty large. But what happens when we add our enzyme? So when we add our enzyme, look at the difference. Our activation energy is much lower now, right? So the activation energy, or the EA, is the energy required to reach the transition state. And so a lot of times what we call this, another term for EA, is delta G double dagger. It is the same thing as activation energy, but it's the energy required to reach that transition state. And so it's the difference in energy between the reactant ground state and the transition ground state. So make sure you know the difference between the two. So here's kind of an example of how that works. This is catalase. Catalase is awesome because it provides a 10 to the 15-fold enhancement of hydrogen peroxide decomposition. Hydrogen peroxide is really dangerous for cells. So the quicker we can break it down inside of our cells, the better. So how does it do this? Well, catalase has a highly reactive iron porphyrin ring inside of it. And so when hydrogen peroxide is bound there, it gets sequestered into this protective environment. And so if you look at the delta G value, right, we know whether we're the normal reaction or we're catalyzed, our delta G is not going to change. So our delta G for our reactions are exactly the same, whether you are catalyzed or not. But what's different is our activation energy, or our delta G double dagger. And so if you look, the uncatalyzed reaction has an activation energy of 71, positive 71 kilojoules per mole. That's quite a bit. But our catalyzed reaction is only positive 8. So this is why we can use enzymes to speed up reactions. So enzymes don't always work alone. It's a team effort. Enzymes can have cofactors. Cofactors are small molecules that aid in the catalytic reaction. And so if you have an enzyme that usually has a cofactor, if it is in the presence of the cofactor, we call it a holo enzyme, and that means it's active. If it is without the cofactor, then we call it an apo enzyme. And the apo enzyme is the one that is inactive. Okay. All right, so there are ions that usually participate and do this. I'll show you a table in a minute. But iron, copper, magnesium, zinc is another really big one. Zinc, if you've got molecular biology as a class, or even genetics, right? You know that zinc is a cofactor that's required in a lot of nucleic acid metabolic enzymes. Right, like transcription factors, all kinds of stuff like that. All right, so you can have cofactors or you can have coenzymes. The difference between a cofactor and a coenzyme is that in a coenzyme, these are cofactors that require organic components. So this can be a loose association, or it can be a very tight association, so one or the other. And so a lot of times you'll hear this with NAD and FAD, right? Nicotinamide, adenine dinucleotide, and flavin, adenine dinucleotide. We'll talk about those. And then you can have a third group that is a prosthetic group. So this is a coenzyme that is permanently associated with the enzyme. So it's part of the entire complex. So here are our cofactors. Remember, our cofactors were the very first category here. And so there are cofactors that are specific for specific kinds of reactions, right? And how they function, their role. You don't have to memorize this. We'll talk about some examples. And so you do need to know the examples, but you don't have to memorize the table. It's not what I'm going for. So let's look at NAD plus and NADH as our example, right? This is really important for things like a lactate dehydrogenase enzyme. So this is one that's important in cells because it converts pyruvate to lactate under anaerobic conditions. So if you're in low O2 conditions, or if you have a cell that doesn't have a mitochondria. I know, right? Doesn't have a mitochondria? What? A red blood cell doesn't have a mitochondria. So either one, right, you're going to convert pyruvate to lactate. So when pyruvate's converted to lactate, NADH is oxidized into NAD+. And so the NADH will bind to the enzyme near the active site, and it's going to function as an electron carrier. And that carrier can then donate a pair of electrons to the redox reaction, and that results in reduction of our enzyme substrate. But then guess what? The cell can recycle it. It's pretty awesome. And the NAD plus that's produced is then used by another enzymatic reaction in the cell. And it's used as a cofactor again, a coenzyme. And it accepts a pair of electrons, right, in the redox reaction. And then that oxidizes the substrate. So they're recycled. We have reactions that require either the NAD plus form or the NADH form. And so they can constantly be regenerated, right? So that's a really a cool mechanism how that works. Some coenzymes are covalently linked to amino acid functional groups in enzymes. And so they serve as integral components in a catalytic reaction. Lipinide is a covalently attached coenzyme, and it plays a role in decarboxylase reactions. And so this is glycine decarboxylase enzyme. And so here's the active site, right, of our enzyme. And so inside of that active site, we have lipoamide, which is our coenzyme, right? And it's covalently linked here, and it sits inside of the active site to help to facilitate that reaction from happening, right? And so you can have both the oxidized and the reduced form, just as we had NAD plus and NADH, and different enzymes can use either the reduced or the oxidized form, depending on what they need, right? All right, so we kind of have a method that we use to name enzymes, and it's a pretty good, I think it's a pretty good method. Most enzymes, like we've talked about before, are going to end in ASE. And we tend to put the substrate in the name of the enzyme, so what it basically does. And so in addition to getting a name, the International Union of Biochemistry and Molecular Biology classification system also gives enzymes numbers based on what they do and how they do it. We're not gonna, I don't care that you understand the nomenclature in terms of numbering. I do care that you can look at the name and you can start to understand what that enzyme is doing. And so usually it's going to give you a substrate and it's going to give you a function, right? So think about a transferase. So it's going to name the substrate, and then it's going to say it's a transferase. Well, what do you think is going to happen? It's going to transfer a functional group, and the name might actually include the functional group, and it might not. An isomerase, right, is going to do a molecular rearrangement of groups. It's not going to take away or add anything. It's going to rearrange it. So it's important that you kind of look at these different names of these groups and understand that you're going to get a function out of it. It's not a structure, so be very careful about that. It is not a structure, because you can have things that evolve independently to do the same function, and so they have different structures, but similar functions. And so the naming for enzymes is not based on structure, but it is based on function. So enzyme nomenclature, this is big time based on function, not structure. It's kind of an important note. Okay, and that is the end of 7.1.